All right, today we are going to look at genetics, and specifically we're looking at Punnett squares. Now I know last week I mentioned a little bit about Punnett squares, and today we're going to look at how can you actually perform a Punnett square. The ultimate questions which we are looking at is how do we get the genes and traits that we do? Where do they come from? And how can we map them so we know who we got them from? So the all traits that we do, and how can we map our genes and traits? Figuring out who we got them from. Did we get them from our mom or did we get them from our dad? Very first thing to look at is what is a Punnett square? What does that mean? Well, as the word describes, it's a square. Imagine that, okay? Now, it's a grid that allows you to predict the kind of traits that an offspring could have. It's like a probability square. You cross them and you can see what your traits will be based on the genotypes that are given. And you know that once you know your genotype, you can figure out your phenotype, which are the physical characteristics that relate to each genotype, okay? So once again, it's a grid that involves your genotypes. So real quickly, we're going to look at the difference between homozygous and heterozygous. Homozygous, as we talked about, is when a trait, a genotype, is exactly the same. That means you have to have the same letter. It has to be uppercase, both have to be uppercase, or both have to be lowercase. If they are not, then they are not homozygous. They are heterozygous. Now it is polygous that are shown, and that would mean it's heterozygous because they're not exactly the same. Okay? So homozygous is the same, exactly the same, and heterozygous is different. It's a mixture. And you can see that here. Homozygous would be the yellow bean plant because they're both lowercase g, and they're both G, whereas the green bean plant is heterozygous. Okay, now we're going to look at Punnett squares and exactly how in the world you can do a Punnett square. So your first step is to write down the genotypes of both parents. Now I picked random letters here. There was no purpose in the letters I picked, okay? For our sake, let's just imagine that codes for a red flower. So we have two parents that both code for a red flower, okay? Now your next step is to write down the genotypes of the gametes they each produce. So that just basically means break them apart. You have two letters, so break each set of letters apart. So you have 4. Okay, so I broke down the capital R and lowercase r. Right. I did the same thing with the other one as well. Your next step is to draw a square. You're going to draw one square and then split it up into 4. Okay, then you're going to draw this Punnett square. So we have it here, a blank Punnett square. And you're going to write each split genotype on the top. So we have the ones written on the top, split, capital R, lowercase r. And you're going to take the second parent. So one set of alleles from one parent goes on the top, and then one set goes on the side. So what you do is you cross the very first one on the side with the very first one on the top. You get capital R, capital R. Then you cross the very first one on the side with the second one on the top. And you get capital R, lowercase r. Then you do the same thing with the one on the very bottom, the allele on the top. You take the second one and cross it with the uppercase r. So you get uppercase R, lowercase r. Then you do it with the second one on the top, lowercase r to lowercase r. So it's basically a crossing game, and you always start with the alleles on the side, and you just make sure you cross the top allele, very, very top of the square, and then you cross the allele on the bottom of the side with both alleles on the top of the square. Something to remember here, though, is that if you end up with an uppercase and a lowercase group, the uppercase letter always goes first. It's just kind of like a rule. We have all those rules in math. This is the same rule in science. And that ends it. That's exactly how you do a Punnett square. You just cross it, cross your genotypes, and you'll get 4 sets of alleles that might be an offspring. Okay, so once again, we looked at what is a Punnett square exactly and how do you work it. If you need more help, go through this once again, just kind of in the alleles in the square. And then you can go on to the bonus question. And that is, if your dad has brown hair given by the alleles, capital B, and your mom has blonde hair given by the alleles, lowercase b, what would be the Punnett square for their child? And what is the probability of the child having brown hair versus blonde hair? Remember, brown hair is genotype. In blonde hair, you must have 2 recessive, so 2 lowercase genotypes. Okay, that ends the podcast. Let me know if you need any help at all. Go through the extra resources on the Ed Canvas site. That will definitely help you.